Ever wondered how real-time PCR works? This clever little technique, an essential tool in the field of molecular biology, is like a magnifying glass for DNA. It allows us to amplify specific regions of DNA, making it much easier to study and analyze. And the real-time part? That's where it gets even more exciting. Real-time PCR, also known as quantitative PCR, not only amplifies DNA, but also measures the amount of DNA amplified during the process, all in real time. This makes it an invaluable tool in various fields such as disease diagnosis, genetic research, forensic science, and much more. It's like watching a thrilling play unfold, where the DNA is the main character and the enzymes are the supporting cast, all under the watchful eye of the real-time PCR machine. So, are you ready to unravel the mystery of real-time PCR? Let's dive in. The first major step in real-time PCR is amplification. This is where we take our double-stranded DNA and prepare it for replication. This process can be broken down into 3 stages: denaturation, annealing, and extension. We start with denaturation. Picture a tightly wound rope. To untangle it, we need to apply heat. Similarly, high-temperature incubation is used to melt our double-stranded DNA into single strands and loosen secondary structures in single-stranded DNA. The highest temperature that the DNA polymerase, the enzyme that synthesizes DNA, can withstand is typically used, usually around 95 degrees Celsius. If the DNA has a high GC content, we might need to increase the denaturation time. Next, we move on to annealing. During this phase, complementary sequences have an opportunity to hybridize or bind together. To ensure this happens, we use a temperature that's based on the calculated melting temperature, or TM, of the primers. Think of primers as the starting point for DNA synthesis. We generally set the temperature 5 degrees Celsius below the TM of the primer. Finally, we have extension at temperatures between 70 to 72 degrees Celsius. The activity of the DNA polymerase is optimal, and primer extension occurs at rates of up to 100 bases per second. That's like adding 100 building blocks to our DNA strand every second. When the amplicon or segment of DNA that we're replicating is small in real-time PCR, this step is often combined with the annealing step, using 60 degrees Celsius as the temperature. So there you have it. Through the amplification process, we've managed to replicate our DNA. Temperature control is key in each stage, influencing how effectively our DNA is prepared, primers bind, and new DNA strands are synthesized. With amplification, we've successfully replicated our DNA. But how do we detect it? Detection, the second crucial step, is where real-time PCR truly shines. In this phase, the magic of fluorescence technology comes into play. But what is fluorescence? Picture this. It's like a glowing marker that lights up under certain conditions. In real-time PCR, this glowing marker is added to our DNA sample. Now, this is where it gets interesting. The specimen, adorned with our fluorescent marker, is placed in a well and subjected to a thermal cycle, just like in normal PCR. But here's the twist. The machine used in real-time PCR is subjected to a light source. This isn't just any light source, it's a tungsten or halogen source. This specific light source is what causes our fluorescent marker to shine. As the light interacts with the marker, it begins to fluoresce, lighting up our DNA sample. And as the thermal cycle progresses, the DNA in our sample begins to multiply. This is the amplification step we discussed earlier. As the number of DNA copies increase, so does the fluorescence. The result, the signal is amplified along with the amplification of the DNA. This emitted signal is then detected by a very special component, the detector. The detector captures the glowing signal and transforms it into digital data. This data is then sent to a computer where it is displayed on a screen. It's like watching the DNA amplify in real time, hence the name real-time PCR. But there's a catch. The signal can only be detected when it surpasses the threshold level. This is the lowest detection level of the detector. It's like a line in the sand that the signal must cross to be detected. This threshold level is crucial in ensuring the accuracy and reliability of our results. And voila! The amplified DNA is detected, and the results are ready for analysis. Just like that, with the help of fluorescence technology, tungsten or halogen light sources, and a detector, we can observe the amplification of DNA in real time. This is the beauty of real-time PCR. Now that we've understood the steps, let's explore how real-time PCR works in a real-life scenario. Picture this. You're a genetic researcher studying a rare genetic disorder. You've identified a mutation that you believe could be the cause, and you need to confirm its presence in patient DNA samples. Here's where real-time PCR comes into play. You design specific primers for the mutated sequence and add them to the DNA samples. The amplification phase begins, where the DNA is denatured, annealed, and extended. This process repeats, amplifying the specific DNA sequence of interest. Simultaneously, the detection phase is underway. Each time the target sequence is amplified, a fluorescent signal is emitted. This signal is detected and converted into a digital format, providing a real-time visual representation of the amplification process. As the cycle continues, the fluorescent signal crosses a threshold level, confirming the presence of the mutation in the sample. With this example, we can see how real-time PCR brings theory to life. We've come a long way, haven't we? From understanding the intricacies of real-time PCR to delving into its two critical steps, amplification and detection. We've seen how amplification is a dance of temperature and time, beginning with denaturation, where DNA is melted into single strands at high temperatures. Then comes annealing, where complementary sequences hybridize at a temperature just below the melting point of the primers. Finally, extension happens at optimal DNA polymerase activity, leading to primer extension at impressive rates. We've also explored detection, where fluorescence technology comes into play. The specimen is subjected to a thermal cycle, and a fluorescent marker added to the sample brightens under a tungsten or halogen source. As the DNA sample amplifies, so does the signal, which is detected, digitized, and displayed on screen. Real-time PCR is not just a process, but an elegant symphony of science. And that, my friends, is the power and beauty of real-time PCR. Until next time, keep exploring and keep learning.